Today's presentation is QSP and Simcyp Designer, Seamless and User-Friendly Integration of QSP and PBPK Models. Our speakers today are Dr. Mark Lovern, Dr. Pete Vandegraff, Dr. Andrzej Kirchek, and Dr. Tariq Abdullah. Mark is currently a Senior Vice President of Commercial and Product Strategy in our Simcyp division at Certara. Pete is a Senior Vice President of Quantitative Systems Pharmacology at Certara. Andrzej is the head of systems modeling in our Simcyp division, and Tariq is an associate principal scientist at Certara's Simcyp division. Welcome to the presentation, everybody. The floor is yours, Mark and Pete.
Thanks, Suzanne. We're really excited to talk to everyone today, and I'm going to start off with chatting with Pete a little bit. Pete, what can you tell us about the origin of QSP Designer? What really motivated its development?
Hi, Mark. Thanks for having me in this webinar. So the idea for QSP Designer first came up some 7 years ago when we launched our first QSP consortia. Now, at the time, this was a novel concept which involved a dozen pharmaceutical companies working collaboratively to develop QSP platforms for immunogenicity and immuno-oncology. Now, we soon had more than 100 scientists involved from a variety of backgrounds and disciplines, and it became quickly apparent that there was no software available dedicated to the development of such large-scale QSP platform models with the involvement of a variety of different experts and stakeholders. So we decided to develop this software ourselves with input from our consortium members. Now, this last point is really important because we basically asked our pharma partners for their wish list of what an ideal QSP software should look like and do. Now, within a year, we had the first prototype, and then we continued this iterative process of releasing new versions based on user wish lists and feedback for 6 years. Now, the result is QSP Designer, which I suppose, based on what I just said, can be best described as QSP software designed by drug developers. Until recently, QSP Designer was only available to our Certara QSP consortium members, but we were starting to get a lot of interest from others who heard about it. So we have now decided to make it widely available.
Excellent. So tell me a little bit more about the collaboration and particularly the wish list. Like, what features did the consortium members and your partners, what is it that they found most valuable and compelling?
Yeah, that's a great question. So, of course, with so many different companies and people involved, we started with a large number of individual wish lists. But what was quite remarkable was the overlap and consistency in terms of the top priorities. In other words, there was general consensus what was most important. Now, I think there were two kinds of wish lists. The first one was more technical and based on input from QSP modelers. Now, one example of a feature that everyone wanted to be included is automated unit checking, which can really save a lot of time and effort and makes model building much less prone to errors and mistakes. Now, Andre will show many more of such technical features in the demo that comes next. So let me focus a little bit more on the second wish list type, which was based on inputs not only from QSP modelers, but also from non-technical stakeholders. Now, broadly speaking, they asked for three main features that are really the foundation of QSP Designer. First, a graphical interface that allows for close and continuous involvement of non-modelers, such as biologists and clinicians, in model building and development. In essence, a QSP model is a representation of biology, and QSP Designer treats it exactly like that. As you'll see in the demo, the graphical representation of the model, also known as biological map, is fully integrated in the software and is drawn like a biologist would draw it. Equations are developed automatically in the background and are not displayed unless you want to. So this makes the model building process much more intuitive and collaborative. A second feature that was a high priority was model reusability and exportability. and specifically a request to make the model agnostic to the specific model coding language. A common concern was that long-term return on investments in QSP were highly dependent on the popularity of a particular software or coding language, which we all know can change over time. So we decided to address this important issue by allowing for model exports to various popular formats such as R, MATLAB, and Julia. Importantly, if over time another model language emerges in the QSP discipline, we can easily incorporate that. So QSP Designer, in essence, makes the model future-proof. This feature also hugely facilitates regulatory submissions of models in a format agencies are familiar with. So to that point, Andre and I are actually running a workshop with QSP Designer at FDA next week. Now, the third feature on the wish list was seamless integration of QSP models with PBPK, physiologically based pharmacokinetics, which allows companies to leverage the investments they are making in these currently separate areas. Now, in the second part of today's workshop, Tarek will demonstrate how we have broken down the technical challenges of doing and how integrating PBPK and QSP models is now literally a few clicks away in our new Simcyp Designer platform. So I look forward to the demos from Andre and Tarek, and look forward to the questions and feedback from our audience today.
Thanks, Pete. It sounds like a very exciting new technology, and I, like you, looking forward to the demonstrations today. And Andre, with that said, if you would start us off on a tour of QSP Designer, please. You're muted.
Thank you, Mark. Thank you very much. I hope that you can hear me well and that you can see my screen. So QSP Designer.
Andre, you're not sharing.
Say again?
Oh, you just need to share the. There you go. Perfect. Thank you.
Okay. Thank you very much. So let me start over again. So QSP Designer is a software for building, calibrating, and simulating QSP model with a user-friendly graphical interface. So the user interacts with the model through biological process map, which you can see here. This map contains all the symbols which are required to define state variables, to define parameters, compartments, and also reactions which contain rate laws. Once biological process map is completed and all rate laws are created, and the software compiles automatically this representation to ordinary differential equation model, which can be also independently examined. While once ODE system is ready, you can automatically create it. You can run simulation. Here you see simulation with one parameter vector for let's say one virtual patient. You can examine all quantities in this model, and you can also overplot experimental data. Furthermore, if you want to plot it in a different software, you can export CSV file, you can get Excel with plots, you can also save a binary database. We actually very rarely run just one instance of the model. What we do usually, we run virtual trials where we point the software to the CSV file that contains parameter inputs for different virtual patients. And then we run these virtual patients one by one. This is happening now. So simulation takes a little bit longer. It's completed now. Now again, we can see all quantities, but now what you can see is a spider plot with all virtual patients that have been run. We also provide parameter estimation engine. So I think I have some technical problem. I'm really sorry for that. Can you show my screen again? Can you see my screen again?
Yep, you're sharing, Andrej.
Yeah, we can see
Okay.
your screen.
Okay,
Yeah.
very good. So I'm showing you now our parameter estimation engine. So you can point the software to a data file, which also defines dependent variable. You can define a standard parameter estimation methods, objective functions. Then you define parameters to be estimated with bounds. And then you run parameter estimation. You get interactive plots. You see objective function. You also see how well simulation fits data. So what I'm doing here, I do dose response fitting. So each of these time profiles will fit data for one dose level, but I will get just one parameter vector for which the model reproduces dose response. This is just about to complete. I hope you see that the fit is not changing anymore. Now it's completed. So finally, this model, as all other models that I'm going to show, was built by dragging, dropping, and connecting symbols from this palette, so we can build models. So I'm aware that the features that I presented so far, very basic features, are available in our other tools, including biological process map interfaces and graphical model building. So let me focus now on features of QSP Designer, which are unique to QSP Designer, which you will not find in other tools, and which we are very, very excited about. So first of all, our biological process map is modular. So if I expand all the modules, you see that each module contains some part of the model and exposes this part of the model for connections through very well-defined interfaces. So the first application of this is just to describe model to different audiences who want to see the model at different level of detail. So for example, I can use this representation to describe biological scope to biologists. So I have tumor, which grows, which dies and produces antigens, which are taken up by dendritic cells, taken to local draining lymph node, where T cell response is recruited. T cells come back to tumor microenvironment, kill tumor. This makes more antigens, creates positive feedback. Then tumor can inhibit this process, and we have our drug with certain PK, which is anti-PD-1 antibody, which binds PD-1, inhibits inhibition, and therefore enhances killing. Now, at this stage of the presentation, I'm going to be inevitably asked by a modeler about some mathematics. So for example, what kind of tumor growth inhibition model did you use? So I can now zoom on the relevant module, and then I can dive into detail. I can show the state variable tumor with unit cell, which is automatically verified. I can show that I have growth process with maximal rate mu, but it is capacity limited. Now the tumor is able to recruit this capacity in the process of tumor microenvironment remodeling. I can show formula for kill function, etc. I can present all details. Another application of modularity is to compose model in modular way, especially if different team members are building the model, connecting legacy models or literature models. And here modularity can be taken actually to yet another level. So let us look at the PK module. So if I open PK module, you see that there is very little content here. Basically what happens is that I point the software to another file. So let us see now this file. So if I open this workspace, you see that this is just PK model developed by somebody else, independently validated. It's 2-compartment PK model with dose, injection site, central peripheral compartment. So when I go now back to the main model, what you will see is that basically what we are connecting here to one variable of this PK model, which is central compartment concentration. Now, both workspaces are combined into one ODE system, so I can create any feedbacks between two models, and you can see that the names of important workspace are preceded by the name of the import, so you can use the same parameter names in both files without risking a clash of the names. Now, my colleague Tarek will show you how this is taken to yet another level and how we use exactly the same interface to connect to our other product, Simcyp Simulator, and then create one ODE system containing what we defined here and what is defined in Simcyp Simulator. So let me change the gear now and show you another unique feature of this software, which is automatic generation of the models when we have combinatorial explosion of reactions. So what I'm showing you here is one of the platform models that Pete mentioned. This is our vaccine simulator, which we used in support of mRNA COVID-19 vaccine development. Now, this model has quite detailed representation of MHC2 pathway. So antigens are taken up by dendritic cells, processed, presented, that recruits again T-cell response, which helps B cells to produce antibodies, which then bind antigen. So I will zoom now on MHC2 pathway. What we are doing here, we are modeling antigen uptake, digestion to T-cell epitopes, and binding to several alleles of HLA genes. We model several alleles so we can represent individual patient HLA genotype. In particular, we model 6 alleles, 2DR, 2DP, and 2DQ. Now we allow up to five T-cell epitopes. So overall, we can make 30 different complexes. Now, we could, of course, represent it by creating 30 copies of this diagram here, but that would be laborious, that would be error-prone, and also it wouldn't be a very nice diagram because it would be unnecessarily big. So what we do instead, we introduce this index notation. You have index J with 6 values attached to MHC, which creates 6 variables automatically. They can have different values. Index I creates 5 copies of this variable. And then when we index complex with both indices, we get all 30 species. Reaction doesn't have an index, and this is because reaction object creates all rate laws automatically just by looking at indices of substrates and products, and we can verify it here. So I will look again at ordinary differential equations. You see this is quite a big platform model. I will try to scroll quickly to relevant part. So here you actually see ODEs for all the complexes, and you see that all these ODEs are automatically created once you also have very nice diagram. So as Pete said, we provided model code export. So you click generate script here, and you can create model code export in R. You can create model code export in MATLAB, in R with equations in C, and in Julia. So the fundamental difference, because between what we are doing here and what some other software is doing, is that we create pure language code rather than script with calls to our solver to application programming interface. So if you generate Julia script, you can take it away to another computer where you don't have QSP designer, and then just work with it purely in Julia. So Pete gave motivation for this. So for example, different modelers can use their favorite language to design, create bespoke simulation scripts which are not implemented in QSP Designer. While doing that, they can use all strengths of these languages, specific libraries which are specifically good for certain things, but also they can use all strengths of their background. All people who code, they have their favorite language, and here we can kind of use all of them. And Pete also mentioned that in some cases, clients will ask us to have a model in certain language, and we can deliver this, and regulators may like to have model in open source environment. So this is all from me. Please do not forget that you can get 28-day free evaluation license for QSP Designer by emailing simcyp.support@certara.com. Thank you very much for listening, and back to you, Suzanne.
Thank you. Thanks, Andrzej. I'm going to pass it over to Tariq now.
Hi, everyone. So as Andrzej suggested, I'm going to show you Simcyp Designer. I'm going to take you through what it can do in addition to what Andrzej showed you for QSP Designer. And so, really, as was suggested, these two products share a common code base and kind of work in a very similar way. However, Simcyp Designer allows you to add these user-defined QSP and PD models to existing Simcyp PBPK models. And so this allows you to extend the capabilities of Simcyp beyond those that are available. And obviously Simcyp can already do a whole lot of different things, but there are a number of extensions that you may think of adding, and I'll show you some examples. As Andrzej showed, you can rapidly prototype the models using the same graphical interface and connect to the predicted concentrations. So this, in a way, gives you the best of both worlds, because you have the flexibility to create and change the models, but you have the bedrock of existing Simcyp models and compounds and population libraries that you can build on and make predictions with. So to just give that intuitively, then, if Simcyp is a house, then Simcyp Designer, is for creating extensions, or potentially replacing parts of the model, or creating more detailed submodels of particular tissues or organs. And it can also be used for creating new routes of entry. So this would be, new routes of administration, or defining absorption into different tissues, in ways that are different than currently available. And so, as you might imagine, that has a whole range of potential applications. You can create sort of much more mechanistic PD models on one level, but then you can also, simulate drug-drug interactions with the existing DDI models, or you can define new mechanistic DDI models. And you can simulate genotypic differences along the lines that Andrzej was suggesting. So if you have parameters in your QSP model that differ for different genotypes, then that is something you can make a prediction with. But I'll focus on one particular example, just because it shows it fairly simply what you can do, which is a case of altered physiology. So, you know, and this is just hemodialysis. So, you know, for patients who have chronic renal failure, you know, the dialysis is then used to filter the blood over some particular period of time when the treatment is applied. You know, and if we look at that a little bit closely, then you see you have blood flowing in one direction through the dialyzer and dialysate flowing in the other, and you simply have a filtration across you know, a membrane. And, you know, really that's happening in lots of little tubes. You know, but on a kind of macro level, you have diffusion across the membrane and you know, also a movement of fluid. So then if we, I will show you now how to kind of apply that within Simcyp Designer. So as you can see, the layout looks, you know, very much like the QSP designer that Andres showed. The main difference is you now have a tab for objects coming from Simcyp, and you can drag on, you know, initial assignments, which can be used to call in parameter values for your compound or your population. You know, and you can also pull in compartments, which can then be linked to you know, the relevant volumes in your model. You know, and lastly, you can drag on the species that you want to associate with particular concentrations in Simcyp. You know, so here we want to access just the systemic concentration in a minimal PBPK model. So we just select it from the list of available concentrations and click apply. You know, and then we would simply, you know, give this an appropriate name, and then, you know, begin to build up our model in exactly the same fashion as Andres showed. You know, and I've, you know, already defined this beforehand here. You know, but you can see the reactions into this dialyzer model are fairly simple. So you just have the blood flow through the dialyzer coming from systemic concentration, you know, and a return which accounts for the fluid removal, you know, and then you have a cumulative amount removed, you know, and we can look in more detail to also see what is going on inside that model in terms of the diffusion and convection. And then, you know, we're applying this dialysis for some particular period of time just by, you know, disabling the reactions with events. So then, you know, we will click run, and what we'll observe is that, you know, this new model has an effect on the clearance of the compound during this dialysis period, which helps us to, you know, account for this effect, which we otherwise are not able to do. You know, and furthermore, you can then take that model and import, you know, different workspaces into it with different compound files, and then, you know, access both existing compound files in Simcyp, such as fluconazole, or, you know, you can go through the normal process of defining new compound files that you're interested in. And then you can obviously look at other things on top of that, such as predicting drug-drug interactions and so on. You know, and with it being in Simcyp Designer, you know, you can make changes to reflect the hemodialysis setup as appropriate. And so I will also just sort of hint at a slightly more complex example that incorporates you know, multiple things. You know, so this is kind of similar to what Andres was showing, but you can, for example, add you know, T cells within your existing PBPK model, and then make a prediction of the, incorporate a model that accounts for the migration of T cells to your tumor, and then, be predicting kind of the exposure of your drug in the tumor, using existing Simcyp PBPK models. But then on top of that, also account for the, the binding to the T cells and the formation of a trimer within Simcyp Designer, and then model the effect on tumor growth. And that is based on this paper. And so as you might imagine, then we can kind of take models that have been developed at different stages, and link them, and reuse them. So at different stages in development. So during discovery, PBPK models may be being developed in animal models, and designer can be linked to the animal models as well as the human model. Separately, people may be using QSP to develop QSP models or quantum system toxicology models. And potentially then you can make predictions in human by linking the PBPK models to QSP models, and adjusting parameters as necessary for the human. And at these early stages, we'll have animal data, so we can learn and confirm from that to update the models as necessary, and make predictions for humans, and then in early clinical development, we can then continue to learn and confirm to improve the models in human, as well as making predictions in special populations, which can then be verified in late development. And there's the potential to share and reuse models across these different stages. So as I've hopefully sort of shown, this allows for very fast model creation. It makes it easy to apply these models to different workspaces by pulling in population covariates and drug parameters, which then update with the workspace. And it has, of course, the same advantages of unit conversion and dimensionality checking, which means less time debugging models and more time designing and applying them, which is what we all want to be spending our time doing. Lastly, I would just indicate that there is a workshop coming up. It's before the PAGE conference. So if you happen to be going to that, you may want to look into this satellite workshop for Simcyp Designer. Thank you.
All right, thanks so much, Tariq. And we're going to pass it over to Mark and Pete to wrap it up.
Well, first, yeah, we're going to do some Q&A. We have some questions from the audience, and then I'll get Pete's thoughts on our future directions for the product. But first, let's hear from some of our audience members. So first question, in addition to the GUI, is there a way to develop the model programmatically using code?
So that the answer is no and yes. So no, we do not have our own scripting language for model development, but yes, because within graphical user interface, you can define rules generating models with indices. So I demonstrated to you how you could create MHC2 binding network, and this is exactly what our partners were doing with the script, creating MATLAB code effectively. Here you can encode such rules with indices, and this could be very complex rules. We can create a full allosteric enzyme model. We can create antibody drug conjugate, PBPK models just by iterating one compartment. Our software engineers have interface to C# where they can script to C# in this way, but this is not yet distributed to users.
Thanks, Andre. So next question in 3 parts, it looks like. First of all, thanks for this fascinating presentation and congratulations on the very nice work. Questions. On exports, why no SBML export? So let's start with that one.
Yeah, so this is a very good question. So I used to be academic before. I worked in a systems biology field since second International Systems Biology Conference in Caltech. So of course I did notice that we don't export SBML. The reason for this was that there was surprisingly low interest from our partners. And another reason for that is that we kind of have a different quality requirements than academic software. I published 3 papers with academic software. We cannot really allow a situation that we will export SBML and then somebody will try to run it in another tool and they come back and say, oh, your SBML doesn't work. And then we will have this uneasy conversation, what went wrong where we are talking about so many third-party things. So that's why we generate our own code in popular mathematical modeling environments, which give us more quality control. Having said that, now that the software is mature, we may get our arms twisted and do it both in export and import way.
Okay, thank you. A couple more parts to that person's question. Part 2, conflicts seem to be managed via variable scoping, but if all variables are scoped, then the two models then do not talk to each other. How do we control then for connecting variables? That's a little, yeah. Yeah.
Mark, that question is actually quite clear. So the scope is controlled by connecting variables and parameters to reactions. So if you have a reaction object, everything which is connected to it is within scope of this formula, and you would have predictive text help showing you scope. If you define parameter and leave it unconnected or assigned but not connected downstream, it becomes global parameter. So depending on your coding habit, you can use local parameters, you can use global parameters. Now, when you have workspace model in 2 workspaces, then of course you have to import a parameter from another workspace, but you can make it global within your main file. And you have all niceties again of object-oriented notation. So for example, in a specific file, you have a control whether quantities are private or whether they are exposed to another workspace to see them. Plus you get dependency graphs within the software. So whatever you are used to in object-oriented programming in terms of scoping, you will find graphical language equivalent within QSP Designer.
Okay, thanks, Andre. I think you may have already answered part 3 with your response, but I'm going to read it anyway in case I misinterpreted, which is, if the same variable is actually 2 different IDs and 2 different models, how do we manage alignment?
Yeah, so if 2 members, if 2 modelers will want to use the name K1 in 2 workspaces and then merge them, then in the workspace, which is in the main workspace, when you call another one, the parameter will be referred as import name dot K1, so you will not have a name conflict. And also, when you connect workspaces, you have to specify these imports. So there will be no conflict, and you will have again control of what is exposed, what is not exposed, what is private. Yeah.
Okay, thank you. Is there any documentation or tutorial that we can read or watch for learning how to use QSP Designer? If so, do we need to have a license to access it? And I think, well, we're giving away evaluation licenses anyway right now, so that's not a problem. But what about the documentation or tutorials?
Yeah. Okay, so there will be tutorials coming with evaluation license. If you look at our tutorial paper in CPT PSP, it will give you a paper describing motivation, describing a lot of things that we were talking about, but also you will find supplementary material which contains detailed technical documentation. I mean, it is not exactly getting started guide, it is full technical documentation. In terms of getting started, there will be a video associated with CPT PSP paper, but also you will get getting started material together and sample models together with evaluation version.
Okay. All right, I'm going to direct this next one to Tarek because it's about Simcyp Designer. And it says, can PBPK Designer allow the user to modify existing Simcyp PBPK models?
So yes, absolutely. You know, provided that you can define the new models as reactions to existing states in the PBPK model, you can add any kind of new functionality that you want to.
Okay, great. Thanks, Tarek. Here's another Simcyp Designer question. You've shown that QSP Designer can be integrated with Simcyp to create QSP PBPK models. How does code export to R, Julia, etc. work in this instance? Does it export only the code describing the QSP model or also the describing the PBPK model?
Right, so in fact, the export to code is not available in Simcyp Designer. That's a feature for QSP Designer only.
Yeah, and the reason for that, let me elaborate, is that we do not export Simcyp PBPK model. So this is why this feature is not available in Simcyp Designer.
Okay. All right, another Simcyp Designer question, Tarek, which is, can Simcyp Designer be used for drug combination PD simulation, especially pulling drug concentrations from multiple compound files? Ooh.
Yes, sure, it can do that. So you can access the concentrations for different compound files in your simulation and then create a synergistic PD effect with it. Sure.
Great. All right, so can we edit the differential equations after generating them?
So no, I mean, again, no and yes. So you cannot go to ODE panel, and in the ODE panel where I was showing you all these LaTeX formulas, you cannot edit it there. What you could do, you could export the code and try to edit in the code, but this is not recommended. The way you edit ODEs is by editing rate laws in reaction objects and editing assignment formula in assignment objects. If you would really like to cheat it, you could create state variable object, assign one reaction having a whole ODE, but this is again not recommended. The reason why we use bipartite variable reaction notation is that you have to type in rate law only once. So once you type in rate law into reaction object, it will be added with plus or minus sign, depending on whether variable is substrate or product in all ODEs where it has to be. So we think that this way of editing ODE system is actually less error prone.
Okay. All right, now we've got a question about performance. In general, for a typical model and runtimes, can QSP Designer run locally on a processor, or does it need to be connected to the grid with multiple CPUs for functional use?
Yeah. So QSP Designer runs as a desktop application. It runs locally. So if we want to run it in high performance computing environment, we have virtual machines with multiple processors. Now, if your computer, no matter whether it's a laptop or whether it's a virtual machine, if it has multiple cores, a lot of simulation algorithms within QSP Designer will use all the cores. So in particular, virtual trial simulation that I was showing you is trivially parallelized. So basically, QSP Designer will fill all cores with parallel virtual patient simulations and will use your computer to the full.
So do you personally, Andre, do you usually run it on your personal machine, or are you running it on a grid usually?
So I run it most of the time on the laptop. The presentation that I was doing with some simulations today, this was run on my laptop. When I want to compute something big, then I connect to virtual machine, I transfer the workspace, and I run simulation there. Yeah.
Perfect. Thank you for explaining that. I think that's probably very helpful. Can we explain the between subject variability using in QSP PBPK using Simcyp? I think the answer to that is probably yes, right? It's like once you've specified the model, you can specify the variability. Is that right, Tarek?
So sure. I mean, you know, obviously the variability in the PK will come from Simcyp, from you know, the population covariates generated in Simcyp and all the population variability generated there. You can then also obviously generate variability on the parameters in your new model that you've defined. You know, where you can define the parameters in the new model as functions of parameters from Simcyp, and in that way also account for the variability.
And the next question was similar, but it was about sensitivity analysis. So I think the answer would still be, you know, the same, right? It's like you can still do sensitivity analysis of the model using the in the Simcyp framework, right, or the platform. Oh, okay. Sorry. Go ahead, Tarek. Sorry.
Sorry, was that for Andrew? Sorry.
Let me read the question as written. Is it possible to perform tasks such as sensitivity analysis in Simcyp Designer? And I don't think it's in the Designer itself. It would be in the Simcyp platform, but using the model built in Designer, right?
So can I take it?
Yes, of course.
Okay. So in the version of QSP Designer that you will receive as a demo, there is no sensitivity analysis. So what you can do sensitivity analysis by changing parameters and looking at the outputs, or you can do sensitivity analysis by generating this virtual population file with many parameter variants and running all these parameter variants together and looking at the results. In version 2, which we have internally, we already have implementation of Morris method, which we will of course release. Now, you mentioned, Mark, that Simcyp simulator has Morris method implemented and sensitivity analysis, but you kind of cannot run the software from that side. So you cannot run. We decided that software will be operated in Simcyp Designer scenario the software will be operated from a Simcyp designer side, and this does not yet have connection to sensitivity analysis. So the same thing applies, that you could do all sorts of parameter changes, but there is no formal sensitivity analysis yet.
Okay, thank you, Andre. Okay, so our final question of the day is, can one develop a QSP model in the QSP designer and then load it into the Simcyp designer?
Yes. Yes, right. Absolutely. I mean, you develop your model in QSP designer, you play with it, then somebody wants to connect it to Simcyp, they come, you load it to Simcyp designer, create connections, and you go ahead. Yeah.
Excellent. All right. Thank you, everyone, especially Tarek and Andre for fielding these questions from the audience. Wait a minute, we just have one late breaking one. In Simcyp, are all necessary parameter values available for any compound specific and disease specific or virtual patients? Or if we create or using PBPK model for stages of specific diseases, can we input parameters or values?
Right. Okay. So I mean, in general, you know, you would take a parameter coming from Simcyp. You know, let's say it's just something like fraction unbound, which might change with a disease state. You know, or you know, or the abundance of a particular enzyme, which will change with disease states or population. You know, the parameter that will come will depend on the population that you use and the individuals generated by Simcyp. There's no way to directly replace those parameters, but you could change them in Simcyp if you wanted to change them for a particular population or disease state.
Okay, thanks, Tarek. All right, we're going to draw a line under the questions now, because I want to give Pete a little time to tell us about the future directions for QSP/Simcyp designer. So Pete, what's on the horizon?
Well, first of all, I hope that the audience agrees with us that, you know, having seen Andre and Tarek's demos and the Q&A, where basically the pattern was a question, can the software do this? And the answer was yes, right? So I hope they agree that the future of QSP and Simcyp design is very bright, and I honestly think it's going to change the way we approach QSP and PBPK modeling. As Tarek said, focusing much more on application and impact rather than on debugging and error checking. So first of all, we very much are looking forward to receiving further feedback from people once they have had a chance to actually try QSP Designer. Now, as mentioned before, a free trial license can be requested at simcyp.support@certara.com. And our recent tutorial that Andre mentioned that was published last month in CPT PSP contains example case studies and instructional videos. And as you also heard, we'll be running an in-person workshop at the upcoming PAGE meeting in a couple of weeks' time. Now, of course, we'll be continuously developing and improving QSP and Simcyp Designer. And as before, this will be driven by the inputs and requests from users and the needs of drug developers. Now, we'll be launching modules integrated in QSP Designer for disease-specific applications in the near future. For example, in immuno-oncology, neuro, and immunology. Now, last but not least, we are also working on the integration of QSP Designer with our Codex clinical databases through the new Certara AI platform, which will really open the door to what I think is the most promising and exciting next horizon, namely linking QSP models to clinical outcomes. So I think QSP Designer will become much more than another modeling software, and I expect it to develop into a knowledge repository for MMIDD, mechanistic model-informed drug development. Now, of course, finally, none of this would have been possible without my brilliant colleagues at Certara and our pharma partners, and I would like to acknowledge all of them but specifically, of course, Andre and Tarek, but also Richard Matthews, David Hollinshead, and Daniel Morris. Thanks, Mark.
Thanks, Pete. Sounds like some really exciting new developments coming. And I want to thank our audience for all the great questions. I'd like to now hand things over to Suzanne, who's going to tell us about some upcoming webinars and events that people will hopefully join us for.
Great, thanks so much, Mark, and thank you to Pete and Andrej and Tariq for an excellent presentation. Just a couple announcements before we wrap things up. Our next upcoming webinar in the series will be on July 19th. Erica Winter from EQRX will be joining us to share a case study on the use of model-informed drug development in formulation development. You can register for this and all webinars on the Certara website. With that, we'd like to thank our audience for attending today's presentation. This concludes the webinar. Goodbye and have a great day.